Steve. So by young, you mean John Young, not age-wise, right?
Yeah, I do.
Yeah, okay. So I'm going to skip over my intro slides here for this group. I think the only one thing that I wanted to point out that we've observed that maybe people aren't talking about are potential reservoirs in domesticated animals. One thing that we're finding through our collaborators is especially cats, and you guys know from the Bronx Zoo with a tiger that was infected, but the cats seem to be shedding huge amounts of virus in their feces. So if you do have a cat, I would recommend putting on some PPE before you change that litter box. I'm going to skip through this for the sake of time and kind of talk to you about how we ended up getting into coronavirus research. My lab, as Steve mentioned, studies HIV and flu, and had tangentially been doing some work on MERS, but not really a coronavirus lab. But this all started when a postdoc of mine went home to China for the Christmas holidays, Zinyin, and he texted me about the outbreak of pneumonia in the Hubei province in China, and how it's been linked to a novel coronavirus. And I think most virologists could almost immediately appreciate when you hear the words novel coronavirus or novel influenza virus, you kind of perk up and start moving into war footing, just because these are viruses that are usually considered high pandemic potential. So in early January, we mobilized and tried to figure out how to start working on this virus. So at that time, the virus was not available in the US. So we reached out to some collaborators out in China, the same collaborators that Dr. Ho has been working with. So Dr. Yuan at a Hong Kong university was actually the one who first isolated the 2002 SARS coronavirus, and they had a clinical specimen from a Hong Kong patient, and also teamed up with Adolfo Garcia Sastra at Mount Sinai. And the first thing we wanted to do, kind of very complementary to what Nevin talked about, was looking at drug repurposing. This was probably the fastest way that we could have an impact on what was at the time an epidemic and now a pandemic, trying to repurpose existing drugs. We were fortunate enough to have a former colleague and an ongoing collaborator, Arnab Chatterjee, provide what, to my knowledge, is the largest drug repurposing library of about 12,000 known drugs. So these are drugs in clinical phase 1, 2, and 3 are FDA approved. This was a collection that was funded by the Bill and Melinda Gates Foundation several years ago. And so the plan was to have Laura Riva, our postdoc in the lab, essentially put a bunch of automation from our lab and the library in a suitcase and fly to Hong Kong and work with Dr. Yun's team to help them do a high throughput screening. They had the virus going there, they had a nice assay going there, and we knew how to screen in 10,000, 12,000 compounds at a time. Laura has run screens for Ebola, for Zika, for influenza, so she had a lot of screens under her belt and was ready to go when there was a travel restriction put into place. So Laura is of Italian origin, so she would have been quarantined up to 14 days returning back. So we made the call that, yeah, she would probably be able to go, but not be able to return. So that plan was quickly scrapped, and we went to a plan B. And our plan B was trying to do this remotely by iPhone. And so we then quickly reconfigured our team with Zin, Paul De Jesus, this is SF out in Hong Kong, and Laura trying to do all of this remotely. So we packed up our automation and our libraries, and of course, Trying to ship during an epidemic or a pandemic, nothing really ever goes right. Our shipment ended up in Guangzhou, not in Hong Kong. And a lot of these shipments, a lot of these items were on dry ice. So Paul was up in the middle of the night trying to get them to refill with dry ice. And as you can see, my hat's off to FedEx for keeping to their word. And the boxes were received, refilled with dry ice. And this is a picture of one of our pieces of automation that made it from our lab into a BSL-3 hood out in Hong Kong. And then for the next 3 weeks, Laura got up in the middle of the night and used the Chinese equivalent of WhatsApp, WeChat, to coach SF here through the high throughput screening process. And I'll tell you a little bit about the details on that. And if you want to learn a little bit more about what the heroic efforts that Laura and SF went through, the New York Times did a nice piece on them about the backstory of this screen. So developing a high throughput screen from 7,000 miles away, as Nevin alluded to at the time, the Vero cells were the best we had. So this was we used what was a CPE assay. Fortunately, we saw pretty reasonable CPE. This is just a picture of the cells dying after infection. The window was good, but not great. It was about a 2.5-fold in 384 well plate. Under other circumstances, we would have tried to optimize this or look for other readouts, but given the situation, we decided to go ahead and pull the trigger. The library used, what I mentioned, was the reframe library, about 60% investigational new drugs. So these are in phase 1, 2 through clinical trials, about 40% FDA approved, and then the rest are preclinical compounds. 80% of these are not found in other repositioning libraries. And in fact, 40% of them required custom synthesis because they are not available elsewhere. So the assay format was as follows. Compounds were spotted, seeded with 3,000 VOE6 cells challenged with a low MOI of SARS-CoV-2, allowed to replicate for 72 hours, and we measured CPE using just a standard live dead assay. So we initially ran the screen on a small library called the low-pack library, about 1,280 compounds, known drugs. So we used a positive control here that's known to block, that was shown to block endocytosis by Ebola. We took a wild shot in the dark, hoping this would work, and it gave us a nice window when you compare DMSO, non-infected, and our control. So our pilot screen looked good, and so we went ahead and ran the 12,000 compound screen. We saw some degradation of the assay window here, but still got nice separation between the control and the non-infected. And the positive control here had some nice correlation between replicates, although we did anticipate that because of this kind of tail here, that we would be screening through a number of false positives. We took that hit and went ahead. So one thing you could do with this kind of compound screen, because these are drugs for which for most the target is known, we can look at enrichment of certain target classes within the screen. And so this is a little bit of a complicated figure, but I'm going to just talk you through the important thing here. So going left to right is essentially a plot of the left-hand side indicates high activity of a screen, and the right-hand side indicates no activity of a screen. So more bars you see kind of slashed on the left-hand, clustered on the left-hand side, represents drugs of a certain target class that were having activity more than you would expect at a distribution that's provided at random. So I'm giving you an example here of a retinoic receptor agonist, and so you could do some fun things with this, such as looking at chemical epistasis. So this was an experiment that was suggested by one of the reviewers of the paper, and turned out real nice, where you can add an RAR agonist and and you see suppression of the virus. And then if you add an antagonist, you can see a reversal of that activity. So meanwhile, back in La Jolla, we received the virus here in early March, and as Nevin alluded to, getting the virus up and going can be a real challenge. It's a new virus, and I was incredibly proud of a postdoc in the lab, Laura Martin Sancho, who was able to get that virus up and going and scaled up to a point that we could screen. So this is the Washington strain that most people are using, and so not only was she using the virus from that we set up here, she also was able to get a assay going using an orthogonal readout. And so this was a readout. Instead of looking at CPE, she did use the antibody provided by Adolfo Garcia Sastre, where he raised it against actually the N protein from SARS-CoV-1 from 2003. And it had brilliant cross-reactivity between that virus and SARS-CoV-2. And we kid with Adolfo, and we tell him this is when it really pays off that he's a hoarder, and he kept this antibody in his freezer for 15 years, and we were able to use this as an immunofluorescent assay. And so our testing down selection panel went from Vero E6 cells, which then we confirmed using this novel IF assay that Lara set up. We did a single point dose confirmation. We got 100 of those compounds confirmed in this assay, not a highest confirmation rate as we would usually expect, but given kind of the circumstances, we're happy to see this. And obviously I won't have time to go through all of the compounds, but this is a nice little cell map. Most of the compounds could not be mapped intuitively directly onto the viral life cycle. Some of them could, including endosomal proteases. I'll talk about them later. ER-encoded proteins and Golgi proteins. The other classes, including GPCRs, ion channels, and a number of signaling regulators, and Nevin alluded to some of those coming up in his screen also. So the next thing we looked at was dose response. For these drugs to work, they need to be available in the patient at significantly high enough concentration. So we set our threshold at 1 micromolar, ideally less than 500 nanomolar of EC50. So we did this in Vero cells to begin with. So these are the classes of drugs known drugs that confirmed. They broke down into somewhat larger categories, including the GPCRs I mentioned earlier, cysteine proteases, ion channels. Remdesivir did pop out on our screen, and the other one was a HIV reverse transcriptase inhibitor that I'll get to in a second. So again, these are the dose response curves. So the two things we're looking for, IC50s less than 1 micromolar, ideally less than, these are cellular EC50s, ideally less than 500 nanomolars, and also a selectivity index where you have a nice separation between cell toxicity, which is in red, and antiviral efficacy. And so what we find is that when you make the cells stick, they don't do anything well, including supporting viral replication. So this is one of the reasons, even though these are known drugs that are relatively safe in patients, this is one of the reasons we've set that threshold in our assays. These are the top candidates. I won't go through all of them, but as you can see, most of them come from indications that wouldn't necessarily surmise that would be necessarily antivirals, maybe with the exception of the two antivirals here in HIV, reverse transcriptase inhibitor and remdesivir. So we surmise that these are more likely than not targeting host cell proteins. Here's some of the more exciting ones. Clofazimine has a real nice IC50 of 310 nanomolar, good separation. The FDA approved drug. that's being currently for used for the treatment of leprosy. And this is the reverse transcriptase inhibitor. It's a non-nucleosidal reverse transcriptase inhibitor, reasonably good. So our EC50s are about 210 nanomolars, which track to the EC50s of the compound for HIV. And we think that because this is a non-nuke, it can potentially be used in combination with remdesivir. The next thing we did, and as was mentioned in the questions below, we wanted to make sure these weren't Vero E6 type of activities that we're seeing, especially because we're going after host factors. So 18 out of 20 confirmed in one or more cell types. So these are ACE2 expressing 293 cells or HUH7 expressing 293 cells. And you can see almost all of the drugs confirmed in one or both of these cell types. So this is when kind of our high throughput screen fell apart, and we weren't able to screen everything in these secondary assays. So we took 9 of these and looked at various different characterization schemes. So one is looking at synergy with remdesivir. So I'll talk you through this data real quickly. So the black dotted line is what happens when you add suboptimal concentrations of remdesivir. The black line here shows what happens when you add the drug, and then the red line is what happens when you add them in combination, and the dotted line is what you would see, what you would expect if the drugs were just additive. So Hang-Fang A is a endosomal cysteine protease inhibitor. It's a quinolone derivative, but you can see there's some really nice synergy, as with some of these other compounds. And I'll get into what some of these compounds do in a few slides. So Hang-Fang A, as I mentioned, is a endosomal calcium channel inhibitor, likely blocking entry. It's been shown to block Ebola entry through a similar mechanism. And while its potency at 1 micromolar by itself isn't great, we're excited about this because of the potential synergy with remdesivir. Another compound, and we think that this might be working along the lines of what Nevin showed. It's a HTB1. It's a mood disorder compound used for mood disorders. And this is targeting a receptor that's largely expressed in neuronal cell types. And so this could be most likely be driven, this activity most likely be driven by an off-target effect and potentially the sigma receptor, which is a known off-target effect of this molecule. So this is, again, the list of compounds. I want to focus on some of the most potent compounds that we pulled out. And I can get into some of the reasons why these tend to be the most potent in the assay. Our secondary assay upped the MOI and reduced the time of the assay. So we did select for entry inhibitors in the secondary screen. And some of these protease inhibitors and kinase inhibitors, we went on to characterize the mechanism of actions for, looking at various 2 different entry assays. So one is a time of addition assay. And so you can see here that these compounds, if you give them early, either before or with a viral infection, you see inhibition. However, when you release viral infection, when you release the virus and then add the compound, you don't see any antiviral activity. And this is very consistent with VLP studies, as these are lenti-based GFP VLPs that Jeremy Lubin had described previously. We can see that these compounds are inhibiting both the SARS-CoV-2 and MERS VLPs, but not the VSV-G pseudotype VLPs. And Andrew Mezikar at Purdue University tested these against SARS-CoV-2 encoded proteases and showed they weren't active. So we're concluding that these compounds are indeed targeting proteases, or there's one kinase inhibitor, but all of these are protease inhibitors. Here, host-directed proteases that are required for spike maturation and endosomal escape. Some of the most potent ones one is a preclinical compound called ZLVG. It was developed as a cysteine protease inhibitor against staph A, but we think that it's working through an off-target activity against an endosomally encoded protease. 190 nanomolar EC50 in Vero E6. This drops to actually double-digit nanomolar EC50s when you move to human cells. VBY is a phase 0 cathepsin inhibitor that was originally developed for various other indications and not antiviral indications, but again, this is potent at 300 nanomolar, and we see some synergy with remdesivir. So moving on, we were able to work with Adolfo Garcia-Saucer's lab, who were able to make iPS-derived pneumocytes, which he showed can replicate the virus very well. And we were able to take a subset of these drugs and test them on these primary human pneumocytes. And while none of the drugs did as well as remdesivir, we did see substantial reduction of viral replication of 3 of these drugs that we tested, ONO, MDL, and apilimod, and tell you a little bit about what these drugs are doing. ONO is a phase 2 clinical compound. It's a cathepsin K inhibitor, which is consistent with a role in spike maturation in the endosome and viral escape from the endosome, and a nice IC50. And the Cmax of the compound is about 1.6 micromolar at 300 mg QD. And so we think that this compound will have sufficient bioavailability to drive antiviral activity in relevant cell types in the lung. And the other compound that I want to point out to you is a compound called MDL 28170. It's a late-stage preclinical compound. It targets a calcium-dependent cysteine protease, which is non-lysosomal, calpain 1 and 2. But again, we think that this is being driven by off-target activity for endosomal proteases based on our data. And so this was originally developed for treatment of myocardial and neurological diseases, but we think that these off-target activities may warrant looking at this molecule as an antiviral. And so the last piece in our down selection filter that we have right now, we were able to, out of Hong Kong, they were able to secure lung tissue from a biopsy, a patient biopsy, and showed that the virus replicates not extremely potently. So you can see the viral titers aren't fantastic, but we are seeing replication. And we can see apilimod, our most potent compound in the screen, performs relatively well in suppressing replication in these cell types, both when the Hong Kong group looked at intracellular RNA and by plaque acid. So that's our down selection funnel up until this point. We're moving other things through. It's more laborious because we can't do these things by high throughput. But just to share with you what apilimod is, it's a phase 2 clinical compound. It's an inhibitor of a PIK5 kinase, which is a known endocytosis regulator, and this is consistent with the data, the VLP, and the time of addition data that we have. An additional bonus for this is that this molecule, it's an antagonist of IL-12 and IL-23, and a suppression of Th1 function. And as all of you know, the pathology of SARS is driven by an unregulated immune response, and so this molecule may have an added bonus of being able to drive down the immune response while being a very potent antiviral. This is a double-digit nanomolar EC50. The Cmax is 2.5, 2.6 micromolar, so we're well within the bioavailability range bioavailability range of this molecule, and the known cellular EC50 for bone osteogenesis assay is within the realm of what we see for antiviral. And this molecule has also been shown to have antiviral activity against Ebola in vitro. So, of course, I think before we go into clinical trials, we want to see in vivo efficacy in hamsters or mice, and then we also have cats and potentially NHPs lined up. And I think that if we are able to show in vivo proof of concept in those, that's when we pull the trigger. And so this is really kind of my philosophy on repurposing right now. So before the EUA authorization for remdesivir, I think it was reasonable to move things into clinical trials as quickly as possible. But right now, remdesivir is and should be considered the standard of care for the treatment of at least for the suppression of viral replication in SARS-CoV-2 infected patients. And so the bar that we're looking for is to see if we can at least match or exceed the efficacy, the in vivo efficacy of remdesivir. Remdesivir is a very tough drug to synthesize, and so there are cost and availability issues for remdesivir. So if we come close to efficacy but are better on cost and availability, I think that gives us reason to move forward. Remdesivir is an IV drug, so if we can come up with something that's oral or inhalation, that's a bonus. There has been some liver tox signal from remdesivir, and so if we can essentially break a toxicity window for remdesivir, not seeing any kind of those associated toxicities at safe doses, that again would give us reason to move forward. If we can show prophylactic activity, safe prophylactic activity, that's another good reason to move forward. And then finally, combinatorial activity, both in terms of resistance and then also being able to reduce the amount of remdesivir that is required for efficacy, thereby extending the worldwide supply so it does become available to the number of people around the world that are going to likely need it. So I've mentioned all of the folks that did all of this work in really a very, very short period. The one thing I want to point out is that our screening data and other people who have now since run the screen are all available at a website hosted by Scripps, reframedb.org. Our funding sources here, and then I need to give a plug on positions that we're trying to fill. If you want to work in BSL-3 in the morning and then go to the beach 3 minutes away in the afternoon, we have positions open for both technicians and postdocs. So thank you for your time. Happy to take any questions.
Sumit, thank you very much. Super. Very
My
exciting.
pleasure.
Yeah. So I have one quick question, then we should let other people come in. So I was just curious about the synergy, which is kind of unexpected in that there are drugs with completely different modes of action that seem synergistic.
Yes.
So that's interesting.
Yeah, so synergy to me is this black box, right? And so in vitro synergy, I mean, if you look at the HIV cocktail drugs, they're not necessarily synergistic, right? But they still work really well together in vivo. So, I think that synergy comes, and I don't hand waving here, right? Comes from when you hit two parallel pathways, right? If you hit the same pathway, there's redundancy there, right? And so maybe you wouldn't expect synergy if you combine, say, two polymerase inhibitors, you would with different mechanisms of actions, because you're really hitting the same nail twice. But if you start hitting two different nails, maybe that's why you would expect some synergy. I know Nevin has done a lot of work with kind of genetic interactions and synergy. I haven't had a chance to talk to him about it. We don't get together as much as we used to these days. So these are conversations we usually have over beers, but I'd be happy, Nevin, if you want to jump in and throw in your 2 cents there.
Yeah, we hear a lot about thinking with pairs of monoclonal antibodies, of course, where you then have really
Yeah,
interesting, complicated interactions of them binding to different sites on the same protein.
right, right.
think Isri Martinez, you can unmute yourself and ask your question now.
Yes, can I know, like, again, what are the data set that you use for, that you was mentioned at the end?
The data, the website is, let me see if I can, it's called reframedb.org. So it's out of the Scripps Research Institute, so reframedb.org. So that's where all of our screening, so our validated screening data, as well as screens that have been run by other groups using different configurations, same library, different configurations. And I think this is really important. Again, you know, we screened at a certain MOI using a certain cell type at a certain time point. And I think if you start switching cell types, start switching MOIs, you can get slightly different. There's some good overlap between the screens, but there's some new things that are coming out when you start looking at this problem a little bit differently using different conditions. And so it's a nice centralized repository for everyone using this screen, this library to find antiviral compounds.